Commencing with the holy name of Allah Almighty, who is the Master, the Creator, and the Emperor of the whole universe. Me, Mariam Saeed, from Grade 7 Thread, is in front of all you to present a presentation on cells. Introduction to the cell, structure and function. Discovery of cells. The invention of the lens. Robert Hooke, 1665, observed a thin slice of cork, dead plant cells, with a microscope. He described what he observed as little boxes cells. Discovery of cells. Anton van Leeuwenhoek, 1675, was the first person to observe living cells. Microbes. Magnification refers to the microscope's power to increase an object's apparent size. Resolution refers to the microscope's power to show detail clearly. The cell theory. Who developed the cell theory? Mathis Claydon, 1838, concluded that all plants are composed of cells. Theodor Schwann, 1839, concluded that all animals are composed of cells. Rudolf Virchow, 1855, determined that cells came only from other cells. The cell theory. What is cell theory? All living things are composed of one or more cells. Cells are organisms, basic units of structure and function. Cells come only from existing cells. Cell diversity. Size, shape, internal organization. Smaller cells, longer cells, and bigger cells. Cell diversity. Shape. Cells differ widely in shape. Most cells are roughly cuboidal and spherical. Cell diversity. Internal organization. Nucleus contains DNA, which directs the activity of the cell. Organelle, a cell component that performs as a specific function in the cell. Eukaryotics, cells that contain a nucleus and membrane-bound organelles. Prokaryotic, cells that lack nuclei and membrane-bound organelles. Eukaryotics versus prokaryotics. Eukaryotics, animals, plants, fungi, protists, and prokaryotics, bacteria, differ greatly in structure. This is the structure of prokaryotic cell. Structure organization of eukaryotic and prokaryotic cell. The parts of the cell. Each living cell carries out of the task of taking food, transforming food into energy, getting rid of waste, and reproducing. Most eukaryotic cells have three main components: cell membrane, cytoskeleton, and nucleus. Cell membrane structure: phospholipids bilayer with proteins with function as channels markers. And receptors also contains cholesterol, which provides rigidity, functions selectively permeable boundary between the cell and the external environment. Nucleus structure. The nucleus is a sphere that contains another sphere called the nucleus. Function: storage center of cell's DNA, manage cell functions. Cell wall structure: rigid wall made up of cellulose, proteins, and carbohydrates. Function: boundary around the plant cell out of the cell membrane that provides structure and support. Cytoplasm structure: gelatin-like fluid that lies inside the cell membrane. Function: contains salts, minerals, and organic molecules. Surrounds the organelles. Cytoskeleton structure: a network of thin fibrous elements made up of microtubules, hollow tubules, and microfilament threads made up of actin. Function: acts as a support system for organelles, maintain cell shape. Ribosomes structure consists of two subunits made up of protein and RNA. Function: location of the protein synthesis. Endoplasmic reticulum structure: a system of membranous tubules and sacs. Function: intercellular highway, a path along which molecules move from one part to another. Two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Golgi apparatus structure: stacked flat sacs. Function: receives proteins from the rear and distributes them to the other organelles or out of the cell, receiving, processing, packaging, and shipping. Mitochondria structure: folded membrane with an outer membrane. The folds of the inner membrane are called cristae. Function: converts energy stored in food into usable energy for work. Cellular respiration. Lysosomes Structure: spherical organelles that contain hydrochloric enzymes within single membranes. Function: breaks down food particles, invading objects, or worn out of cell parts. Peroxisomes: structure: spherical organelles that contain enzymes within single membranes. Functions: degrade hydrogen peroxide and toxic compound that can be produced during metabolism. Cilia and flagella. Structure: hair-like organelles that extend from the surface of cells. When they are present in large numbers on a cell, they are called cilia. When they are less numerous and longer, they are called flagella. Both organelles are composed of 9 pairs of microtubules arranged around a central pair. Function: cell motility. These are the structure of cilia and flagella. Centrioles: structure composed of 9 sets of triplet microtubules arranged in a rank exist in pairs. Function: centrioles play a major role in cell division, mitosis. Vacuoles: structure a sac of fluid surrounded by a membrane, very large in plants. Function: used for the temporary storage and waste of nutrients and water. Plastids: there are 3 types of plastid in plants. Chloroplasts discussed on next slide. Chloroplasts synthesize and store pigments. Leucoplasts store food such as starches, proteins, and lipids. Chloroplast: structure stacked sacs that contain chlorophyll surrounded by a double membrane. Function: photosynthesis, conversion of light energy to chemical energy stored in a bonds of glucose. This is the secretory pathway. Plant cells versus animal cells. Plant cells are very similar to plant cells except for the following major difference. Animal cells do not contain chloroplasts. Animal cells are not surrounded by cell walls. The vacuoles in plants are much larger and those are of the animals. This is the structure of animal cell. This is the structure of plant cell. Hope you like it. This was all from my side. Stay home, stay safe.